Here at ASMS, I gave a presentation on translational workflows, which are workflows that we're developing at the BRIM Center, which are designed to address all the aspects of proteomics, going all the way from discovery of new biomarkers and then taking those directly into more routine applications using mass spectrometry. The workflows can be separated into two parts, discovery and targeted. For the discovery workflows, we typically are using our high-resolution instruments, and we are doing a broad-based fishing expedition to look for new biomarkers in complex clinical samples. Typically, we can find hundreds or even thousands of proteins when we look at these samples, and the data are extremely rich. Some of the workflows that we've designed allow you to look at these proteins with respect to their abundance. If you have different types of samples, let's say a research sample, a test sample, and a disease sample, if you look at the proteins that are differentially abundant in those samples, it gives you an indication of what proteins might be important to the particular condition. Then the trick is, how do you turn that type of an assay or that type of a process into a more routine process where you would want to measure and quantify those proteins in hundreds and hundreds of samples. And that is more of a verification type of workflow that is much more routine. And again, at BRIMS, we've created workflows that can take you all the way from that discovery point through translation into the targeted assays, so that you potentially could now take these into a clinical research environment and quantitate hundreds and hundreds and hundreds of these samples with these particular proteins that are of clinical importance very, very quickly. It's taken, I think, the field of proteomics and mass spectrometry in general several years to really pull together all the technology and all the knowledge to be able to do this complete workflow. And it's taking us into the age of personalized medicine, where we can now screen hundreds and hundreds of different types of proteins in a particular person or related to a particular disease and quantify those very accurately. We just recently published a paper on insulin. One of the key challenges that many of the pharmaceutical companies have when they're giving these therapeutic analogs is actually measuring them in the blood of people who are taking them. It was, up until now, not really possible to measure in a single assay a number of these therapeutic analogs, as well as looking at the endogenous insulin that is being produced by the person. So what we did was we developed an assay on our Q-Exactive mass spectrometer. It's a high-resolution, accurate mass targeted assay, and we're using a particular type of enrichment that's called MESA, which is basically an immunoenrichment. We take an antibody to the insulin protein, and we bind it to a column, and then we put the sample through that, and we enrich for the endogenous insulin and for all of the therapeutic analogs. And because we are able to enrich in this way, we can now put it on the mass spectrometer and very accurately measure and quantify all of these different therapeutic analogs. Someone in a clinic could then say, okay, my patient is on this particular type of therapeutic insulin, and the levels are X, and that tells me that perhaps that dosage should be adjusted, or perhaps that's exactly the correct dosage. This is actually something that could be tremendously useful to patients with type 2 diabetes, and hopefully help to improve their day-to-day health. All of the assays and workflows that I'm talking about are strictly in a research environment. None of these types of things are actually being implemented right now in clinics or hospitals, but in a research-only environment, they're extremely helpful in pushing the boundaries of what we can do and what we can find out and how we can help people.